You're watching Coronavirus in Context. I'm Dr. John Whyte, Chief Medical Officer at WebMD. Today, I'm joined by my friend and colleague, Dr. Eric Topol, the founder and director of the Scripps Research Translational Institute and an avid tweeter. Eric, thanks for joining me.
Oh, it's always good to be with you, John. Thanks.
I call you an avid tweeter because your Twitter feed is terrific. You succinctly give us the latest data, the latest evidence about where we are with COVID. But, we were talking right before we started about how often, sadly, there's a lot of negative responses to what you're presenting as facts and data.
All right. Thanks, John. I've been on Twitter for over a decade. So I've actually had a chance, almost 11 years now, kind of see the trends, at least, from my perspective. And until the pandemic, there was a little bit of anti-science that I would see in response to posts. But what happened during these months in 2020 has been nothing short of outrageous, horrific. Mm-hmm. Basically every day, I get attacked for facts, truth, evidence. Many days I thought, John, I just better stop. But, I use Twitter as a way to archive what I read. Mm-hmm. So, years ago, it became my main archival means of tracking the things that I want to look back at. So I might just go back and do that without Twitter. I don't know, but it's been challenging.
I wanted to get your thoughts, just because you're deep in the data on all of this, on where your best guess is on treatment. And, even on your criticisms on convalescent plasma, we've seen the same issues in terms of remdesivir and others. Since when do we change outcomes, Eric, at the end of a study design that we're saying we're looking at death, and no, we're not looking at death anymore. Now we're looking at time to leave the hospital. How is that good science?
It's not good science. When we make antibodies from COVID-19, we make lots of antibodies, not just IgM, but of course IgG. And it turns out that when you look in depth at these antibodies, many clones any of us would make, most of them have no neutralizing capacity.
Right.
So it's only a tiny fraction in most people that do the job, that take down the virus. So the problem is you give this convalescent plasma to a person who's sick, and most of the antibodies they're going to get are not going to do anything. Now, unfortunately, it's such a great altruistic thing. I've been sick, I've had an infection, I want to help other people, and it's been, of course, nurtured. And that part's really good. The problem is there's this illusion that the plasma infusion is going to have all this potent neutralizing antibody. That's not the case. That's why we have programs of neutralizing antibodies that are very potent, that are proven, pure, basically ideal. This is a very suboptimal.
Well, ideal, strategy. But also say, if we're injecting monoclonal antibodies, they also can have untoward effects as well. And, also impacting.
So can plasma. Some of the problems that we can't screen out that could be virus pathogens, for example, we may not know that for years, just like what happened with Hep C, and who knows. So the point is that it's not safe. Monoclonal antibodies engineered that are potent, they may not be, but at least it's pure. That is, it's pure IgG neutralizing antibodies, not diluted with a very small fraction. So biologically, this doesn't have legs, that it's going to be saving life in a big proportion. Maybe it'll have a small, modest effect, but that's about as good as we can ask for. Mm-hmm. Now, and it's actually a big deal to get this, convalescent plasma fractionated to patients. It's not like it's it's an expensive and arduous task. Mm-hmm. Other programs, like you saw what hydroxychloroquine did, that was another FDA emergency approval that had to be revoked because of lack of safety and lack of efficacy, months later, pressured by the president with outrageous, aggressive promotion of hydroxychloroquine.
But to be fair, even in good times, Dr. Hamburg, as you know very well, would say, at FDA, you're criticized either way. You either approve a drug too fast or you approve a drug too slow, right?
Yeah.
People could say it's an emergency, right? We're in a pandemic that occurs once a century, Eric. We have to try things.
Right. No, I'm with you, John, and I don't want to hold back anything. I mean,
No, I don't think you are holding back. You should.
yeah, I
Yeah.
just want to make sure that we have a signal
Right.
because hydroxychloroquine we knew could have some cardiac arrhythmia side effects. We knew there were, more cardiac arrests in certain studies, like a doubling, and it wasn't like a slam dunk there.
What regulatory flexibility should they have used? And we should want to use regulatory flexibility during these times. So what was the right mechanism?
The right mechanism would be, in each of these cases, we have insufficient data from randomized trials. There certainly weren't any randomized trials of hydroxychloroquine at the time. Now, the remdesivir was a different story because, as you pointed out, John, aptly, they changed the goalposts from survival in a relatively small trial of 1,000 patients, approximately, to recovery time. And that was significant. And the p-value for survival was 0.06. So there was this trend. But unfortunately, it was not a large enough trial to determine survival. And what was happening after the convalescent plasma, to add insult to injury, here again, the commissioner of FDA issues, we're broadening the indications for remdesivir under emergency approval based on no prospective, meaningful randomized data for small studies.
I didn't think you would be a supporter of data that's trending because you have to have your data points, right? If we always said if something's 0.06, that might not even get published.
Well, I don't think there's any magical p-value, 0.05. The fact that the mortality was reduced to that extent, and the fact that it helped recover, you could say, okay, well, remdesivir helps in severe COVID-19. Okay, and you want more replication, fine. That I can see, but the point about then giving it a broadened indication, essentially by the EUA, to say anyone who goes in the hospital should get remdesivir. This is expensive. It's in short supply. The data are weak, flimsy. I mean, this is just one more thing in this. I don't have the evidence. I don't have the data, but we're going to give approvals for things. Now, your point, I can't emphasize enough, which is in a pandemic, you don't want to withhold anything that could help people. But at the same time, you don't want to promote things that lack evidence. So there's a fine, delicate balance. And, dexamethasone, for example, went through the right trial. Over 6,000 people, improved survival substantially. That's the kind of data
in a certain
we should
patient
have.
population, right?
That's the data we need to be certain, really confident of anything.
Well, let's give some confidence to people now that we've criticized all potential options, Eric. Where do you see therapeutics going? On your radar screen, what do you think has the best chance in terms of?
Well, I mean, I do think there are some good things on the horizon. The first that we touched on were the neutralizing antibodies because they are so potent. And they are good because as soon as you have the diagnosis, you can basically squash the infection. So you wouldn't progress, and they should be helpful even in someone who's in the early phases, not severe.
They won't develop immunity though, correct?
No, but they should be good for at least a few months, which is important in a person who otherwise could be very high risk for morbidity and mortality. So I think but the problem with those is they're relatively expensive. They're not so easy to make at scale. The dream that I talked about with Tony Fauci, I know you did talk with him as well, but that you could actually give it sub-Q, yourself, that we would have it available to all high-risk people if need be, that kind of thing. We're not going to have that, but that would be, the fantasy of a very effective treatment. Now, the other one, which is preventive, and that's what I emphasize here, is the treatments we have so far, dexamethasone and, more modest effect of remdesivir, are late benefits when someone's really sick. We want treatments that prevent this, the toll of the disease of COVID-19. So the one that has the best data right now is interferon. To give people interferon, which, as you know, is the natural innate immunity. It's the first line of defense. It consists of cytokines and interferon. Now, it turns out that this virus, this nasty virus, is very good at deactivating your interferon response. And some people don't have a very good interferon response at all, and particularly with age and male sex and all sorts of risk factors. And some people genetically, even young and healthy, don't have the ideal interferon response. So the idea here is give people an inhaled interferon early.
Yep.
And there are many studies now, both observational and small randomized studies that support that. So I think that will emerge, I hope, as useful. There are many other repurposed drugs out there, but those two, monoclonal antibodies and interferon, basically supplement, if you will, those look the most promising right now. I wish they were going to be definitive evidence like yesterday, but we're still a little bit of ways from that.
Well, let's talk about the vaccine and where you think we are. I want to first address, the elephant in the room of, a clinical hold on AstraZeneca's vaccine development program. That's a good thing though, Eric, isn't it? Isn't that showing the system is working? It was a serious adverse event. Transverse myelitis, not completely uncommon in a vaccine trial. Note it, trial put on hold. Success, in a way, the process is working?
Well, we don't know, John, too many details. We know that in July, there was a temporary hold on the trial. Someone had, a participant had multiple sclerosis. We don't know too much about that, but it was deemed not to be related to vaccine. And now we have basically a cousin of MS with this transverse myelitis, which could be from a virus. It could be from an immune disorder. We don't know. And we don't know it's a woman who's apparently getting much better, but we don't know exactly what was going on in terms of temporal sequence. So fuzzy. But you definitely, until you definitely want to put the trial on hold. And you're right. This is the way it's supposed to work. It does work. I mean, trials all the time have temporary holds, large trials. And you basically wait. You don't enroll anyone else until you really study what happened, because you don't want to have something like that happen again. So yes, we don't know enough right now, but I do think that there are a lot of things about the vaccine trials that it's critical we do know. We don't have the data analysis plans from these companies and these trial lists. These are done by academics, but they are working with companies. We don't have their stopping rules that are known as part of that data analysis plan. It's not been transparent except for what's put on clinicaltrials.gov.
I mean, it's it's common that there's not transparency in development programs, but given a pandemic, we can't be having information parsed out by press releases and abstracts. Everyone has to know the data, both good and bad. As when things aren't approved, typically we don't get the information. But in this situation, wouldn't you agree, we have to really argue for complete transparency?
Right. This is a very delicate matter in many dimensions. For one, as we discussed, we have this FDA foundation trust issue. We have a president who's using this vaccine as a way to promote his election campaign. So we have that in the background. Then we also have the issue of making the call prematurely on a trial, whereby if it's based on a limited number of infections, these are moderate or severe infections, you're extrapolating that to tens of millions of healthy people that you're going to give a vaccine to. And once you stop the trial, I mean, it's like you can't go back. And then, people say, well, that's it. And then you find out potentially bad things later, and then you lose the trust of the entire public, which is already compromised and fragile. So everything's on the line. All the high quality, high velocity science that got us to this,
which we should celebrate, right? Look at the innovation that we've had to get to this point of phase 3. What's your prediction though, Eric? When do you think, based on, your knowledge of the process as well as what's going on, that we would have a drug for, and I'm going to, parse words for the audience, true approval, not an emergency use authorization. So full approval based on phase 3, probably with a phase 4, you'd agree, post-market surveillance. When's that time frame?
I think we'll see it if we let things do naturally in the first part of the new year, anytime in that first quarter, perhaps, ideally, with enough confidence that the efficacy is solid and that we haven't run into, any significant adverse, serious, proportionate adverse reaction. It's impossible to have zero adverse events.
Sure.
The hope is that it's
No
exceptionally
serious
low.
adverse events.
Yeah, I mean, I think once you treat 100 million people around the world or billions of people, you're going to see antibody-enhanced immune reactions or immunopathologic reactions. I mean, we're going to likely see that, but hopefully it's at 0.000 whatever percent incidence. I want to see this thing where you don't interrupt a natural experiment that you design. Because if you stop early, the chances that you have stopped it at a point of exaggerated benefit, right?
Mm-hmm. What are you optimistic about?
I'm actually optimistic about everything. You wouldn't know
Doesn't
it.
sound like it.
No, I'm optimistic we'll
Yeah.
have a vaccine. It'll be safe with those caveats I mentioned and effective if we are patient. I'm optimistic we will have, other repurposed drugs or new monoclonal antibodies. I'm optimistic that with the right leadership, we will model and get more people in the country to use the things that the measures that we need to do, like wearing masks and trying to keep distance and avoiding crowds and, all that sort of stuff. So if we get things on track, we can prevail over this. I'm confident of that, but we've been very impatient, very divided, in terms of this, to start, as you started, the animus and anti-science. We have got to get much better education and buy-in of the public that, things like masks and vaccines are good. Right now, we have people who are not believing that.
Well, we don't want to be anti-science. That's what we started with. But we also don't want to be sloppy science too, as we've talked about in terms of data analysis. Dr. Topol, I want to thank you for sharing your insights. I want to thank you for helping educate us here today, as well as every day that you do on Twitter. And we're going to check in with you if we can in a couple months to see where we are in the latest data around vaccines.
Thank you, John. I'm always happy to talk to you, and I look forward to more interactions.